It'll zoom in a little bit. I'm just worried if I use this suction, it's really gonna like break it up. But
Yeah, it might be the best option though. It's okay if the test gets a little bit crushed, as long as it's like, what we really want to avoid is the like impaling.
Like a
right.
little
It's a
worm.
yeah, little polychaetes hanging out.
Come back,
Looks
Bob.
like the same associate as we found with the halothurians, actually.
Cool.
That little polychaete
Yeah,
swimming
can you
around
make a
by
note of
it.
that? Cool. Yeah, the polychaete's like on the urchin.
Yeah, they were there when we found it.
He's pushing the urchin away.
Oh, it's helping.
Current's not helping me right there. All right, I'll go for the suction then.
Yeah.
You want to use the other arm to keep the urchin in place when you push against it?
Yeah, I don't know.
Right, you have
It's
to
a
be
broom.
really...
We'll go for the,
Yeah.
we'll try the suction, I think.
Okay. I'll bring it back in.
Sure. Actually, that's good right there for a sec. Nice sitting on a little marble or something. Okay, let's do,
I'll start for like 10 or something. Probably need more though.
let's just bump it up like 5 at a time, but yeah, let's zoom in a little bit, please.
All right, I'll start off with 5.
Yeah, hold on. I'm just going to change my heading here.
Let me
I'm sitting on a weird rock here. I need to just adjust. Let's come wide. It's too wiggly.
Good thing the urchin's not running away. Like the other one did. That was fun.
Yeah.
Oh yeah, there's a big rock behind me there. Which way should I come around? I think I might come around this way.
Oh, I see the rock now.
You can actually see the urchin in the Atalanta view.
I know.
Yeah. That's really cool. That's a nice shot.
Oops.
Uh-oh.
All right,
No harm.
reload. Okay, so let's start slow, bump it up by 5 at a time.
5, 10. About the make contact. 15.
Maybe another 5.
20.
I need another 5.
25.
Heavy.
Oh, there we go.
Okay, we'll come wide, please. And then open the drawer. Nope.
Nope.
I need to go one more 5.
Copy. You're right there and 30. You want to rack back?
Oh yeah, rack the camera all the way back. Great, thank you.
So a backhanded frisbee golf shot this time.
Little baby hook.
Careful with that handle.
Yeah.
And
Can you give me bubble?
Polychaete's going with it.
Yeah.
oh, hey.
Okay, let's turn it off.
All right, shut down.
I like the dust vortex in the background.
That was good.
Cool.
Nice job, Josh.
Beautiful.
Okay. Close the door.
Very
And
nice,
that
thank
was
you.
sample 161.
That's the
Thank
way it's
you.
supposed to go.
Right.
Juggling routine.
It's not as cool as the
So was
the
that
basket.
35 in the end that you were using?
35, yeah.
Cool.
Wow.
Is that-
30,
Oh,
30.
30.
30, okay. Yeah, they're basically completely full of water, so that's why they're so neutrally buoyant. There's like almost nothing to them but water.
All right, fully wrecked
Spines
back.
floating by.
Nice.
All right, that was nice. That was smooth. That was
Yeah.
good.
That. See if I can get this without the stick in the. No, oh well.
Ready for the ship?
Yeah.
Bridge nav.
Yeah.
Can we go 270 at 0.3 knots for 50 meters?
Yeah.
Perfect.
Tomorrow's Friday.
And you want to come around to port, right?
Yeah.
Another urchin down.
Boom.
Are you about done with the urchins now?
No, I like them.
You like them?
Yeah.
That's good.
All right.
They're-
Always a challenge.
You're not afraid of a challenge?
Yeah, I feel like they're that perfect balance of like delicate, but not squishy, so you can't easily
Yeah.
slurp or pick it up. Crinoid.
Okay. All right, everyone, we've got some questions coming in, so let us know when you're ready. It's pretty much for all of our scientists, navigators, and pilots. So one question that I'll address real quickly: when we start new expeditions, does everyone share their preferred pronouns during orientation? We are given the option to provide our pronouns when we're submitting our applications, and then we do list those pronouns on our bios, and we do share them during our orientations and introductions. So thank you for asking that question. There's another question here for the pilots. Let me think here. Does the Nautilus have plans to work and explore cold waters in the future?
Not in the near future, no. I think we're going to be staying in the South Pacific area for the next few years.
Oh, okay.
Yeah, sort of between Hawaii,
Right.
Guam, and Samoa kind of areas.
Oh, fun.
Well, we're in pretty cold water now, though.
It is
Yeah.
pretty cold water.
chilly
The
down here.
temperature
It's
is about
usually
2
in cold
degrees.
water.
Yeah,
We are in the
Herc's
cold
in cold water.
water. Don't. Okay.
We're in warm water.
And in regards to our communication between scientists, so we do have a group of scientists aboard the ship with us during the expedition, as well as a collection of scientists that are on shore that we communicate between to during our research and data collection and species observations, or organism observations, excuse me. The other question is, how did you decide how to label the collection drawers?
They've been labeled all kinds of funny things over the
Yeah,
over
someone
the
was.
years. So lately it's been this Greek letters and stuff. But
Oh, okay. They were wondering why one was listed Omega and the others were ABC. So it's a combination of both of those. It sounds like.
Probably don't want to get your boxes and slurps confused, right?
Yep. Yep, you don't want to get them
that's
confused.
true. As long as they're different, it doesn't really matter.
Thank you. And then for our navigator, what's the shortest distance you can move the ship?
I don't know. The shortest distance I have moved the ship is 5 meters. So
In some cases, we've moved it like 3 meters. And
Yeah?
Stuff. Yeah. So I don't know if they
Precision
can actually do
handling.
that sort of resolution, but
yeah.
That's when we want to get really close to the cliff.
Up on the bridge, they're like, yeah, sure.
yeah, they
Okay.
just punch the numbers in and something happens, I guess.
Well, I still remember on one of my first cruises before we actually had GPS. That's how long ago it was.
Oh my gosh.
And I was up on the bridge with the captain and just, watching things and chatting. And one of the chief scientists called up a half a degree course change. And so I said, that's cool. So captain didn't do anything. And he goes, no, we can't do that.
What is that off in the distance there? Oh, maybe it could be just a spot.
Oh, sorry. Oh, I don't know why I'm apologizing to you.
It's Canada Day tomorrow.
You
Oh,
have
so I'm just like in the spirit.
26 minutes.
Another anemone.
Everybody repeat after me. Oh, sorry.
Oh,
Sorry.
Sorry.
Sorry. Is that it? What is it?
It's stabilizing a bit more, so that's good. It's just sometimes at certain angles it just doesn't want to change.
Yeah.
It just doesn't want to go.
It's been pretty good for the watch. I mean, other than the big heaves that kind of mess it all up.
Yeah.
I like the different patterns of the ripples,
yeah.
like right in the rocks there, they just kind of give that like half a rainbow kind of, and then they start to ripple out. It's really neat. Like these are pretty.
Ah.
What?
Huh?
I thought you said, ah.
Oh, no.
Bridge nav, we can move the ship at 270 for 50 meters at 0.3 knots.
That was probably the last urchin on this geo.
I really doubt that.
What?
It is like the scale of it is pretty phenomenal. Like the area that we're looking at compared to the size is
Oh my
nothing,
gosh.
right? It's,
It's
like
so
a pin,
little.
It's a pinprick compared
Yeah, I
to like
know. I can you even
huge
see it
feature.
in the scape of the
I know. It's like, wow.
Yeah.
Massive
Imagine
feature.
if all these flat-topped ones were like islands on a point.
Well, they were, right?
I know, but like, wouldn't that be cool now? Now we just have Johnston.
Yes.
And the little ones.
Just.
Just.
I got to explore, opportunity to explore a submerging seamount where that was still the very, very top of the ring of the seamount was just breaking the water.
Really?
And then the caldera area was about maybe only 60, 80 meters deep.
Whoa.
And it was just a massive coral reef. Like it was super cool. Whoa. But it was out in the middle of the ocean.
That's amazing.
Wow.
Yeah.
Did ships ever run aground there? Because I didn't.
No.
No?
So, not that I know of anyway. You could take the whole ship. It was so big. It was like this kind of thing. It was so big you could take the ship right inside the
Really?
top
Wow.
of the seamount. Yeah.
Whoa.
Where was this again? Here?
That was in the Timor Sea, so it's kind of in between northwest Australia and
Wow.
Indonesia.
Oh, nice.
Was that the one with the sharks in it?
It had tons of sharks,
Oh,
sea snakes. It had all kinds of cool stuff.
I heard about that.
Fish and
It's called
Atalanta.
Scott Reef.
Scott Reef?
Scott Reef, yeah.
Oh.
I was working with AIMS, the Australian
Good
Marine.
job.
I'm coming sideways. What does that stand for? AIMS? It's just basically the Australian sort of
Cool.
Marine office. Yeah, it was amazing. That's just like kind of what I envisioned this would have been like,
Yeah.
I guess, you know, all those millions of years ago, just like a teeming like
That's so cool.
coral reef area.
Is it hard if you can't, I'm assuming in places like that where it's just like a bunch of coral reefs, you don't want to land anywhere on anything?
No, we never really landed, no. It was we were using a small ROV. It was really shallow, right? Like only 60 meters, so we were just using a little ROV. But yeah, everything was gigantic. It was crazy. The sponges were huge.
Oh my gosh.
Corals were huge.
Okay, so I have a question coming in the chat here for Liz. Deep sea coral aren't symbiotic, right? So it shouldn't be affected by bleaching.
Yeah, that's correct. Bleaching happens when corals in shallow waters expel their zooxanthellae, which are the little photosynthetic microorganisms that live inside their tissues. Because there's no light down here, these corals don't have any photosynthetic zooxanthellae, and so they would still be affected by warming and acidification, but they wouldn't necessarily bleach.
Okay, thank you, Liz.
Mm-hmm.
bridge nav, we can do another 50-meter step.
Lots of different ripples here. Interesting.
Right? How do they, is it just the situations of the rocks that kind of cause those different ripples to have that pattern where it looks like they're in different directions?
It looks like it's
and
channeling the water,
chunks,
the flow
yeah.
somehow.
That's cool. Oh yeah, of course. Sun? Oh. Yeah, no problem. Oh, here's the languages too.
Fish friend.
Fish friend?
Oh yeah, looks like another cutthroat eel.
It's funny how the species and animals we keep seeing change so drastically from place to place and like landscape to landscape.
Yeah.
Yeah, it seems like each dive site has like had a...
It's like a different signature animal.
Right. That's a good way to put it.
Yeah, I mean, it's part of this cruise is trying to look at what we call biogeography of,
Yeah,
you know, why
kinda
is this
see.
community here? And then at this other site that might be at the same depth, why is it a different community?
Right. Well, it's nice to have you and Rob with us so then we can kind of understand it from both perspectives, from the geological perspective and then the biological perspective.
Right. I mean, I think geology and biology work together so well.
Right.
Like, you know, geology drives what biology exists where, but then also a lot of geology is derived from biology. Like, you know, that sediment there, it's all used to be animal, or not animals, but microorganisms.
But it also pulls in the chemistry and the physics too.
Yeah.
It's really, really a beautifully cross-disciplinary, interdisciplinary sort of.
Yeah, I love being in
Right.
oceanography for that reason. I have to think about all those different things.
Yeah, I kind of understand the purpose and function of all of them working together.
What do we have here? Oh, I think that was a sponge stalk. The yellow is interesting.
It's a crinoid, maybe? Oh, zoom in, please.
Oh yeah, it is a crinoid.
Oh, and a little shrimp.
Yeah.
And an umbellula in the background.
Where's a malula?
Up here.
Oh, that one thing again. Oh, what is that?
It's a seapen.
Thank you. I was thinking it was that little... I keep thinking they're like cauliflowers, but they look like
the
sand.
xenophyophore? Yeah,
Yeah.
looks like it could be.
Yeah. From a distance without my glasses on.
I wonder what awesome skeet we're going to hand over to.
Why is no one wearing their glasses on this ship?
Yeah.
I always forget them. I don't know why. Well, they're mine are sunglasses, so I wouldn't be able to see really good.
You don't wear your sunglasses at night?
I didn't know. No, not at all. Oh, that song. Oh.
How cool would it be to have your comprehensives be a dive watch.
That would have been a
Oh
lot
my
more fun
gosh, that would be
than what it
great.
was.
Did you get through all of that? Or
Yeah.
you're pretty much powered through and now you're just finishing up the good stuff?
Yeah.
Awesome. I heard that that's the hardest part.
It was pretty rough.
But that's awesome that you did it.
My trick was I waited too late in my degree to take them, so they didn't dare fail me.
Automatic pass.
Bridge, now we can do another 50-meter step at 0.3 knots.
What's the master source right now for the
The,
So Yvonne, maybe this might be a question for you.
yeah,
Have a
master
look at
position source? What's the selections on that one? Right now it's on dead reckoning. Oh yeah, that's right. Okay, that's what we want.
why?
I don't know, I'm just wondering why the autos are not always engaging every time.
Oh.
But you have it on, it's right over there.
Hoku, what is the question?
Somebody was asking if there's a shallow water 90-foot max place like these that could be scuba dived on. Really curious to see the early changes. I'm assuming it's within the monument, but I would assume no. But
well, NOAA has done a lot of dives around the reefs around Johnston Atoll, pretty close to where the surface area is. Yeah.
Oh, nice. Okay.
But I just don't have the level of knowledge about what's been done out there.
Right. Oh, thank you. But there has been studies at those depths. Cool.
Yes.
Going about 5 minutes left in our watch.
Oh, hey. You're muted. Is going to get muted in about 5 minutes. If you have any more questions for us, please pop them in the chat. We're happy to address them. Thank you for all your comments and information that you're providing about this deep sea environment. It's very helpful.
And after this dive, I think we're going to begin to head to Johnston Atoll proper and do some work there.
Sweet.
That'll be our shallow dive, right?
Yeah.
Cool. Very exciting. So please follow us on our website. We're going to have more updated information that will let you folks know what our next dive will be, where and when.
But stay tuned. I think this will last at least another 2.5 hours.
Oh yeah, so we will still be exploring this this area for quite some time. So yeah, stay tuned, keep watching, and pop those questions in the chat.
Maybe we'll see more crazy jellyfish.
Ooh, and some sharks. What was that?
Bit of detritus.
Thank you.
Yeah,
Every time I see that detritus fly by, I think it's something extra special.
What? You don't think detritus is special?
It is special. It has this very important. It's very
let's
important.
zoom on the sponge.
Want to zoom the sponge?
Yeah. Sorry, I wasn't paying attention.
That's okay.
It's also a shrimp floating.
Yeah, so this dive is going to last for a few more hours. It is still the same site as yesterday. We had the
Say that,
opportunity to just spend more time in this place. So that's why you're seeing us moving about and exploring this area much more than we had anticipated. So yeah, we'll be here for a few more hours.
Look at mine, please.
It should be smart enough to like go the speed that you are going. That'd be cool.
What's up?
Oh, if it could just continue the course, like in speed, right when you hit it.
Yeah, well, you can set the speed
Mm-hmm.
and then your step size, but it's for whatever reason, it's not taken every time.
Bridge nav, we can do another 50-meter step.
Couple of
What?
well, I thought that was a seapen, but it's like an old stalk or something.
I think
down
that's
there.
a live sponge.
Yeah. Oh, it is. Okay, small.
Yeah.
So we have one final question for our pilots.
Sure.
What is the purpose of the 2 green lasers?
Sure, yeah. So those are the way we use scale, and so they're 10 centimeters apart, and then we can use those to scale anything that we see on there, sizes of rocks or animals, so we can measure things.
Thank you, Josh.
Yeah.
And those 2 lasers sit 10 centimeters apart?
Yeah.
So then gives
Zoom
us...
in, please.
Oh. What is that?
Whoa.
Oh, that's another tube anemone.
Yeah, that's cool.
Oh,
Wow,
cool.
I had no, I could.
Yeah,
That looked
those
like
are
so many
very
different
cheap.
things before I figured out what it was.
So you can see then the laser dots there, it's about 10 centimeters wide.
It's like burgundy too. It's really pretty.
I like those.
Yeah, me too.
Yeah, I like those too. Really cool.
Nice shots there.
Yeah,
Looking wide.
it's cool to be able to see the tube
Mm-hmm.
with
Well,
all the sand on it.
Yeah, yeah, how they use the sand to make that.
Yeah, I like it.
Oh. So our final question is, what does my name mean? So Hoku in Hawaiian means star. It also is a phase of our full moon. So 2 full moon phases, one is Hoku and one is Mahealani. Yeah, thank you for asking. It means star.
Cucumber. Oh.
All right, mahalo nui for spending time with your muted crew. We are officially muting ourselves until we meet again. Malama pono, ahui ho.
Bridge nav, 100 meters, 270.
Hello, everybody. Welcome to the 12 to 4 Team Pickles, Sea Pickles, with a Z. We are currently cruising along on top of a geo. It's sandy with some ferromanganese encrusted rocks here and there. Should see a bunch of fish, some sea pens.
Yeah.
Maybe a shark if we're lucky.
There is a sea pen.
I see a sea pen to the left and a fish to the right.
Shaw.
Hence why I just said that. Not very original. The plan right now is to sail, cruise along for another 2 hours, 2 hours and 15 minutes or so, and then pull up so we can be back on deck at 4.
Roger that.
Little Japanese sand garden over here.
Right where we left it.
Just a little overview with where we're at for samples. It looks like we have 2 Niskin bottles left and 1 slurp jar left. And in boxes A through D, there's only 1 rock. So I don't know if y'all want to double up on those, but that's what we have available at this time.
Thank you, appreciate it.
Roger.
More rocks.
Good scientist Meredith says there's no reason to leave those Niskin bottles empty before we leave the bottom. And Meredith, who's going to have to process water at 4 a.m., says, do I really need 2 more samples?
I agree, let's fill them with rocks. Just pop it right next to some sediment, see if we suck them in.
We can just pop it and then hold the top open and use the scoop and the arm to scoop.
Making all the friends.
Getting creative, I like this.
Can we get a zoom on that sea pen? Or fish?
Fish, yeah.
Yeah, fish.
Fudge.
Shadow is playing tricks on me.
Go ahead and push on there, please. Halosaur.
Yeah, what fish is this?
My money's on the halosaur, hey?
I think this is a halosaur, yeah.
Go ahead and get a little more in there. Do one by down.
Yeah, this is a halosaur. Let's have that head that's slightly different color.
Go ahead and come wide, please.
So Kevin, even though we're up at the top of this geo, we're not on the summit. We're still, looks like we're moving up several different inclines, or at least along one incline.
Yeah, it's a very, very shallow slope, but the highest point is to the southwest, so it still does go up a little bit.
So are we seeing what we're potentially going to be seeing for the next couple hours, or?
Yeah, I don't expect anything different. There's a little cone to the southwest, but I think it's about like a few hundred meters. It's too far. I mean, by 100 meters, I mean like 1.5 kilometers. So I don't think we can make that. So instead, this is probably what we're going to see. But there should be some cool stuff up here. There won't be a lot of corals or sponges, but we might see some interesting, hopefully we see some crabs. I would like to see some crabs.
Got a nice little field of sea pens right here.
Yeah. It's kind of interesting to see the sediment here looking all, has so many different angles to it.
Yeah, and you can see how much it gets disturbed by the rocks.
Yeah.
swimmer.
Yep. Yeah, some shadow of the fish. Yeah, it is.
So cusk eel.
Could push on under bit, please. Oh, is that it wasn't, oh, sorry, let me go this wheel.
Is this about the biggest they get, the cusk?
Oh, they can get much bigger than that. Cusk eels can get quite big.
Partial zoom.
Oh, wow. About how much longer do you think?
I have seen video of a couple around hydrothermal vents that look like they're probably about 4 feet long, and I've seen a couple off California about that size. And they get a really big, deep, thick body too.
Yeah. And the shield. You want to put it somewhere else? Yeah, fair enough. Um, 8 hours actually. At least with it in that position, you can see it all the time.
Little tiny polyapogon.
Oh, it's definitely in the shot. I've been told it gets cropped out. I'm gonna flip it around, okay? It does have a wide angle, you know. It's like the hands up against the bumper, yeah? So it's probably on level with the bumper. Sure. Wrist down? I did. How much up do you want though? Because I don't know what the goal is. Is that what you want? No.
That is really interesting, that the tracks on the ground there, I guess they're just waveforms, but you can really see where the water curves around in there.
Yeah, you can definitely see the current's really close to the surface because it's getting deflected pretty strongly by these rocks. But it has I'm trying to remember the name of the waves, like right in the center of the screen, where they overlap. There's a specific term for that, and it represents that current is changing. It's not a constant current. Wish I remember my sedimentology. Oh, there's a tab open, so I'm guessing Rob had the same question.
Oh, sure. Thank you. Thanks for the reminder. Hey, can I get audio levels on everybody when you have a moment?
Yeah, I was just about to ask that because my alarm went off right next to my ear, so I'm like, am I hearing everything okay?
Ooh, a jelly. Can we get a zoom? That's a cool looking one.
Go ahead and push on in there, please.
Jelly.
Nice jelly.
Beautiful.
I believe this is one of the ones that is bioluminescent.
It's weird that it's floating up and down but not moving at all.
Yeah.
Or at least from our perspective, we can't see it.
Yeah, it's very interesting.
And I call it a puppet jelly.
Oh,
Yay!
Same.
Shows
Wait,
me.
see that?
Gotcha. Gotcha. Is there a way to see if it's bioluminescent right now? Like, can we turn our lights off and see or anything?
You would need actually a low light camera to really detect that.
Oh, that's unfortunate.
Aw, so cute.
We could probably detect it with eyes, but the cameras would have a hard time with it.
I love it so much.
It's coming for us.
Wow. It's coming for those thrusters.
No.
we won't get it in our thrusters, guys. But we will leave it alone now. Goodbye, friend.
Goodbye, friend.
My favorite is one of its like little cut sister taxa. It's a little narrower, that when they get bumped or encounter the ROV, they scoot away really fast. It's very cute.
Admittedly, I probably would too.
Yeah.
Sorry, pull wide.
Yeah, so let's do a round of introductions so we can make sure I can still hear. All right. Hello, I am Onye, your Science Communication Fellow on board the EV Nautilus. We're currently exploring, I guess we're at the top of this geo right now, Wetmore Seamount. And we are, we'll be exploring for about another 2 hours, and then we will be ascending our vehicles. So send in your questions and I'll pass it along to the team.
Hey, all, how's it going? It's Kevin Conrad. I'm a marine geologist from the University of Nevada, Las Vegas. Here to answer any and all questions on geology. And yeah, excited to be here. Don't forget to smash that like and subscribe button. And hit us up on our Patreon. Thank you.
I'm Meredith Everett. I'm a geneticist at NOAA's Northwest Fisheries Science Center, where I work on the genetics of deep sea corals and sponges with NOAA's Deep Sea Coral Research and Technology Program. And I'm here to tell truths about biology and lies about geology.
Hey everyone, I'm Chanel Vidal. I'm a PhD student at Arizona State University, where I'm a geologist turned biologist, and I'm studying microbial life of Antarctic permafrost, and I'm here as an ocean science intern.
Hi, I'm Chris Krasnowski, and I'm currently resetting the DVL. Okay.
Very much appreciated.
And I'm here as a navigator, also high-resolution mapping specialist, and back ashore, I am a postdoctoral researcher at University of Rhode Island.
Hey folks, my name is Jess, and part of the ROV team, flying Herc for part of this evening, and then flying Atalanta for the rest of it. Yeah, like the sea floor.
Yeah, like to see more. Hi, I'm Kylie. I'm sitting in the Atalanta chair. Remember to hit that notification bell.
I don't think any of those buttons actually. Maybe we do have. We can subscribe on the YouTube channel.
Yeah,
Okay.
like, subscribe, and hit that notification bell.
I'm pretty sure there's no Patreon though. Just to clarify. My name's Tim.
Personal Cash App is.
Yeah, you can just Venmo me at kevin.com. No.
Kevin loves geology.
My name is Tim Dwyer. I am sitting in the video engineer chair to Kylie's left, and I'm managing the video feeds and the audio levels and video recordings here. I'm from Friday Harbor, Washington, where I'm a science teacher. Oh, there's not a jelly.
Oh, yeah. It looks like the resting pose, this one, hey?
Yeah.
I lost my hat, but I found it.
It's a very small sombrero.
Go ahead and push it on in there, please.
Do we know what type of species this jellyfish?
This one, I always forget its name, and I am looking it up right now.
Okay, just kidding. Come a little wide there. All right, go ahead and come wide, please.
He's moving.
So are we.
That's right. 2 ships passing in the night.
1 wee ship and 1 big ship.
Yeah.
So this sediment that we're looking at, is it mostly sand or marine snow?
It's going to be almost entirely marine snow.
Cool.
There will be little bits of wind-blown dust. So places like the Gobi Desert and Sahara, they can have that dust during storms blow all the way out into the middle of the ocean. And
Really?
there's some areas of the world where that's the only sedimentation. You get these thick red clay layers where they have super low sedimentation rates, but they build up slowly through time just from this wind-blown, what's called aeolian dust. And you can also have in these sands, you probably have what are called interplanetary dust particles, which are essentially tiny little micrometeorites that settle at a constant rate around the globe.
Wait, there are micrometeorites that settle at a constant rate all over the world?
Yeah, there's essentially constant dust flux from the solar system and the galaxy that deposits on Earth. You can detect it using usually isotopic means. Helium-3 is one of the ways you can look at it, especially if you look at some of these areas where either it's carbonate like this or it's a red clay from wind-blown dust. But anywhere where you have super low sedimentation rate, you can start actually measuring and detecting the interplanetary dust concentrations through isotopic analysis.
I'm actually studying this for one of my secondary projects at ASU.
Cool.
Antarctica is really useful for finding these because, like, it's white there and dust shows up like really well. So we're looking at these micrometeorite particles which fall and people find in Antarctica and determining their isotopic composition and correlating them with like larger meteorite bodies in the, what is that asteroid belt called again between Jupiter and, oh, what is it called? You know,
the Kuiper belt?
Yeah.
No, it's not the asteroid belt, isn't it?
Oh, yeah, just the asteroid belt. Okay. Yeah, well, there's specific meteorites that like already have been characterized that we're correlating our micrometeorites to. So, yeah, and some of them are like pre-solar grains too, so we can like they form before Earth even formed, which is pretty cool. So, yeah, sometimes I spend time looking at rocks from Antarctica, and other times I spend time looking at pre-solar grains from Antarctica. So it's grad school is not too bad.
I think those jellies are in the genus Atolla.
Cool. So I have a paper that just got back. I'm doing revisions on it, so it should be hopefully published soon. But it's on looking at supernova events in sedimentary cores. And so about 5 to 2.5 million years ago, Earth was impacted by a local supernova. And exotic particles from that supernova kind of scattered down on Earth. And you can actually pick them up in sedimentary cores using isotopes like iron-60. And so we were looking and trying to see if it was a actual supernova event or it was a just like a cosmic irradiated meteor shower event or something. And it, by all means, seems to be a supernova event that occurred about 2.5 million years ago. to maybe 3. We got the timing a little more tight now. But yeah, it's amazing that there's a cross-section between marine geology and astrophysics.
That is amazing. I never would have thought that, detectable levels of particles from an exploding star or imploding star would, exist on the surface of the planet.
Yeah, the cool thing about it is there were isotopes that are made when a supernova occurs. So iron-60 is one, but has a half-life of only a few million years. And so on Earth, all iron-60 is gone because Earth is obviously significantly older than the half-life, and so it's decayed away. But we found a pulse of these iron-60 in sediment cores, which means the only thing it can come from is supernova events. Yeah, and then there's some work trying to figure out if it affected different climate shifts that occurred, 2.7 million years ago. I don't know if that's pushing it, but it could affect it.
That's really fascinating, Kevin. That's cool stuff.
Yeah, thank you.
Blowing my mind, middle of the night.
Someone in the chat is wondering, where can we find that paper on that information?
Yeah, so mine won't be published for a little while on the noble gases, but the main paper on iron-60, really well done, is by Walner. It was an Australian group, 2016. Let's see, what was the name of the paper? Recent near-Earth supernova probed by global deposition of interstellar radioactive iron-60. Very simple name. But I think you can get it probably open source. It's a Nature paper, but that's the best one to look at for it. Or just Google Walner supernova event, and you can find some articles written on the paper. That'll probably be a little more friendly if you're not into astrochemistry. Can we get zoom on the seapen?
Sure thing. Go ahead, start your zoom there, please.
See. Yeah, this I think is a seapen called funiculina. There's a couple different species. The most common is quadangularius. You can tell this because the polyps aren't occurring in leaves. They're just loose and individual all over the stipe. And if this is the same one that we collected. It's in the forward box. And when we get it up on deck, the polyps will most likely have a little crown of spines around each polyp.
Cool.
Someone,
sorry. Tim, you want to come full wide for a moment there?
Yeah, someone in the chat is wondering, how exactly do we look up sea creatures? How do we identify them?
Some of it's just experience from some of us who've watched a lot of deep sea dive footage and handled a lot of specimens. And then we also, on the computers in front of us, have a number of computer tabs open to some deep sea guides that have been produced by NOAA and by the Hawaii Undersea Laboratory and some other sources.
Chris, would you mind centering up here? Thank you.
Actually, I think we can pull this off.
Roger.
Meredith, what different groups do deep sea research out here? NOAA and the Hawaii Undersea Research Lab. Who else?
Well, all of us aboard the ship from a number of different institutions. There are, I think there were the two main groups operating out here recently. Would be, NOAA with the Okeanos, and then Nautilus, and then the Hawaii Undersea Research Lab.
Can you push on there, please, Tim?
I'm not aware of any others, but that doesn't mean they aren't getting permits and ship time.
USGS did a lot of work in the area like 30 years ago, 40 years ago, but they haven't recently.
It's the United States
Hawaii, please.
Geological Survey.
Geological Survey.
Yeah.
Can I ask a silly question? I know you're going to say yes, but I'm not sure where it's going to go.
Please start.
Aside from surveying the geology, what does the USGS do?
Well, they survey the geology. They do baseline mapping. They do a lot of research projects, so various topics of, ranging from climate change to sustainability to a lot of work on mineral resource identification. And yeah, so they do a diverse amount of things. I mean, originally they were just mapping areas that haven't been mapped, which they still do in further detail. But now they have more guided projects that are specific. A lot of focus on earthquake hazards, seismic hazards, volcanic hazards, landslides, stuff like that.
Okay. They'll also characterize whole environments with that kind of focus in mind, but that means they also have teams that work on biology. So I work with a number of biologists who work for USGS and geneticists. Yeah. And so they have a whole lab that's devoted to salmon research and disease in Seattle as well. So they are another research agency that, while their primary focus is kind of landforms and geology, they will use that as a baseline to characterize whole environments, and that includes the biology.
Oh, Raj. Hey, Jess.
What's up?
You've got a little bit of slurp in your frame.
Oh, sorry. Thanks.
No worries.
Thank you.
If there are any students or budding geologists watching, USGS also has an awesome internship program that a lot of my friends went through throughout their undergrad degrees. So they had really great experiences, and a lot of times it translated into jobs at the USGS after their internship. So if you're studying geology or biology or related field, definitely look into it.
Yep.
Would you mind doing a DVO reset there, please, Chris? Did you want to go dead rec instead of USBL for a solution?
So Chris, is the multibeam able to pick up on ripple mark structures? Or the sonar, I should say, multibeam, sorry.
Yeah, it is.
That's interesting.
Maybe not quite this small, I don't know.
Because then you can do like a survey analysis, like machine learning, and reconstruct current directions automatically, which would be really nice instead of looking through the video footage.
Yeah. Let's put the backscatter on and see what we see. I've definitely seen sand ripples in larger sand ripples on some of the surface surveys and things we've done. I mean, if they're big enough, you can just resolve them like in the bathymetry, but if not that, sometimes you can see them in the backscatter. Let's see.
Yeah, can we have a zoom on that floaty thing?
Sure.
You want porch on for this?
It's another jelly.
Sure.
Oh.
A little atolla. My favorite / the most depressing thing ever about some mud dives is when you have fish that are sitting on the mud and you realize you can see them in the sonar because everything else is so flat.
Does it look like it has a yellow part to it?
It does. It's got a little toupee.
Or helmet or hood.
It definitely looks like a different species of atolla.
Get a little tighter zoom if you can.
You know those bike helmets that have like the
little spikes on them?
The spikes, yeah, sticking out. Sort of looks like that.
Yeah, this one looks a little different.
Bit, please.
It's cool.
Has an interesting little pattern on it too.
Yeah.
Do we know what type of shark the previous watch came across?
Given the description they were talking with, I think it was another cat shark like the one we saw on our watch.
Tim, do you mind resetting the zoom again there, please? Thanks. Thank you.
Is it possible to see any type of rays out here?
I think we can. I don't know that there are any deep species of rays in this region. I know in other regions in the Pacific, you'll have deep sea skates that can get this deep.
I'm gonna do a pilot swap here.
So Kevin, to answer your question, if you pull up the Norbit screen, you can. If you look here, I pulled the side scan mode up, and you can see
Channel 3 for those at home.
Yeah, you can see sort of these. You can see the little sand ripples. I mean, you can obviously see the rocks and stuff. Now everything's getting smeared because the vehicle is stopped, but you can see. Yeah. So you can definitely pick them up.
Yeah, that's awesome.
Yeah, side scan is something that I tend to not think about a whole lot because normally I'm doing bathymetric stuff, but some of the side scan things can be pretty cool too.
Yeah. Yeah, I think that would be neat. If you have a whole bunch of data from a cruise, and if you can come up with a program to process it and get currents automatically, that'd be fantastic.
Take a quick zoom.
Yeah.
Oop.
It's a little... Is there something on top of it? I can't tell. I don't think so. I think it's just bent over.
Yeah, I think it's just tilted over.
Okay, come on.
And goes pulling in its polyps.
So Chanel, where do you find all these internships? It sounds like there's quite a few out there for students who are willing to look and apply.
Oh, for sure. There's so many. The big place that I found internships were asking for opportunities for my professors and undergrads. So I really liked a few of my professors and ended up in their office all the time with just random questions and advice on which classes to take. And professors are a great wealth of knowledge, and they can be very intimidating, but they definitely want to hear from you, and they'll have a lot to offer if you come to them with enthusiasm. So that was my #1 stop. I also just Googled internships. Also, Twitter is really active when scientific conferences are going on, and people tweet out opportunities a bunch. So it doesn't seem like Twitter would be a place where you can find professional opportunities, but I know friends who have found like postdocs and other internships. So yeah, speaking to professors who you find interesting, literally just Googling geology internships, Twitter, and also just looking at an organization I would want to work for. So like the USGS, I thought was doing really cool stuff. So just Google USGS internship or National Park Service internship, and pretty much every organization has some way to pull in undergrad students. So yeah, if you want the opportunity, it's probably there. You just need to ask someone to help you find it.
Can we zoom on that little guy?
Mm-hmm.
And yeah, that's a great point. Pretty much every state and
and
federal
zoom.
agency has some internship program. Yep, another one of those atollas. This one is almost all black, or dark colored at least. It's very cool. And very tiny.
Do we know the life cycle of these jellies?
I don't know if they know the full life cycle of these. I know with other, they're known for other medusozoans, and it's likely that these have a very similar life cycle.
Okay, come wide. Get out of here. I like you too much to be close to you. Goodbye, friend.
So most scyphozoans have a free-living medusan stage, which is what we would be looking at, and then they also have a benthic stage where they form little stacks of polyps. So in the adult stage, they'll broadcast spawn their larvae. Those turn into a little grain called a planula that will settle out into the benthos, grow as a polyp for a little while, and then the polyp becomes this little stack of mini jellies that then release one at a time and back into the pelagic stage. And if you Google for the photos of their benthic stage when they are forming their little stacks of mini jellies, it's actually quite cute, and it's quite cute to watch them like release and become these little mini jellies up into the water. There's a couple aquariums that have an exhibit that has all the life stages.
The little guys, the little ones beat so quickly. They're trying so hard.
That pattern's wicked here.
Yeah. Yeah, a lot of cross currents, a lot of localized kind of eddies and stuff. Not
Range
consistent at
300
all.
meters, 270.
Sponge.
Wow, that's cool. Kylie, mind if I push it on Atalanta?
No, yeah, go for it.
You want to push on in there a wee bit there, Tim? Okay. Wow.
I think it's a small fish.
There's two little tiny fish there. One right by the lasers and one just left and below.
Oh, Raj. Can I get a zoom?
Tadpole size.
Wow.
Little guy. It's mesmerizing. Okay, come wide. If I tilt up, it'll be easier to light.
Thanks.
Yeah, no worries.
It's like, it's too hot, too cold.
I know, and let me know if you're like needing me to adjust, because that cue will be helpful for me.
Okay.
Yeah. The chrysogorgia?
Yeah.
And something
looks like metallogorgia
down at the bottom. Was a crinoid maybe?
I couldn't tell what that was. Possibly a crinoid, possibly a rock pen.
Just gotta
there's another one there. It's a black coral.
Oh,
Heteropathes.
Beautiful.
Or bathypathes.
Really?
Yep.
I don't see a lot of corals on the top.
No, those are the first two that I've seen. I've seen some bamboo bases, but not actually anything alive.
What's the difference between the bathypathes and the parent pathes?
Heteropathes? They're two different species. They have very similar morphology.
Okay.
One has slightly shorter polyp limbs and more branchlets that come off the main pinnules. And then just to really confuse things, there's a third genera called alternatipathes that also looks very similar.
There's another lovely jelly.
Okay, take a zoom.
This one's looking less healthy.
Yeah.
Oh, wow.
Look at those things that float around in The Nightmare Before Christmas. I've never seen that movie. I'm not sure if the reference is right. I'm not sure that's right. Okay, come wide.
As I say, I don't remember anything like that.
Maybe I'm confusing that with like Pac-Man or something. Something is wrong in my brain. But we knew that, didn't we? Oh, Jess, tell me when you're going to do that.
Oh, sorry.
It's okay. It turns my brain upside down.
Gotcha. You said that, and now the song is stuck in my head.
Zoom a little bit. So we can get some of that texture. Okay. Let me see.
Nice. It's already shot.
This is pretty.
It's cool.
Yeah, lovely sponge. I don't see an associate in it, but
yeah, me either.
can't really see through the top. Yeah.
Right, yeah. Okay, come wide. I gotta catch up. I'll lose my driving privileges. The sponges all look pretty healthy and tall.
Yeah, they don't have a lot of, they're not completely covered in brittle stars like the other ones we saw earlier.
It seems like on a lot of the dives, like, we're seeing like maybe one-to-one, like, dead sponge for a live sponge. And I don't see too many dead stalks. Have you guys?
No.
Not up on top here.
Yeah.
It's a nice stalked crinoid. I find interesting with these little Euplectellidae vases is they seem to have very much a maximum size. You don't see ones that are just absolutely enormous like you can get with some other species. I wonder if they're just so fragile that if they get too big in the current, they break.
Right. But they can't grow on themselves either because they have that circular structure. Like a polyapogon seems like it can just keep growing on itself and just getting thicker.
Yeah.
This a wolteria?
Yep. Yeah.
Go ahead and zoom.
Kevin's getting really good with the IDs. He might be crossing over into expert biologist realm here.
I will forget every single thing by the next cruise. This is going to be a coral pretty soon.
All kinds of benthic tinaphores all over the fronds there.
What are those little guys under the rocks?
Tighter if you want.
Okay.
Are they anemones?
I'm going to pan out.
Can't see them
wide.
anymore.
Yeah.
Let's get out of here.
You ever find seapen gardens?
Yeah. Oh, yes.
Oh.
I have seen many a seapen garden.
I remember one there was, I don't remember where it was at, but it looked like a bunch of little cattails or squirrel tails or whatever, like popping out of the sediment. It was awesome. I wrote in my journal, I was like, I flew through a forest of cattails. I don't remember the rest of the dive, I just remember that.
I have on a hard drive I've got with me a bunch of photos that some of which are, there's a seapen garden and there's a the hylodyma sponge garden. So they're these sponges that grow in soft sediment on these, they're also called fiber optic sponges on these long stalks of essentially fiber optic fibers. And then it looks like being in a giant garden of crazy tulips.
Oh, cool.
Can we get a zoom on that stalk straight above the oasis?
Straight above, yeah. Just get over here.
That looks like a yellow crinoid. I don't think I've seen one this dive yet.
Oh, I think that's a bamboo coral.
What is that sediment? No, I think that's a crinoid. On the wolteria to the right.
Okay, coming on the zoom, thank you.
This is a bamboo coral that would cause headaches for taxonomy because it looks like it's trying desperately to be a single branch bamboo, but then it has just gone ahead and branched.
It's a sparse brancher.
Yeah.
It kind of looks like my drawing skills, like when you draw a person with the stigmas.
Sorry, I lost my heading.
It's all right, I lost my focus. All right, here we go, zooming in.
Twice, lost it twice.
All good. We can see what's at the base.
Okay, come wide.
Going wide.
Thank you. Ugh.
So Chris, on Norbit, the red area that we're seeing is the rocks that we're coming across, right?
So I have it colored by backscatter now instead of by depth, since this is a really flat area. So the red bits are the bright bits. They're the ones that have the loudest echo. So that generally means hard surfaces, hard or close. So that's why the rocks light up a lot more than the sand.
Do you always use the little Hercules? as the little driving icon.
Yeah, well, so that Hercules is like totally to scale and everything. So that's I kind of use that to keep track of the reference frames for all of the sensors and stuff.
Oh, nice.
So if we zoom in, you can see all these little coordinate frames that are labeled in there. All actually keep track of where everything is. Oh, so like right here shows where the USBL with the Norbit is up here, and in there is the INS. And back here is the DVL. So, yep, this is exported right from the CAD model of Hercules.
Nice.
That shrimp was all natural zoom.
All natural.
Yep.
It's coming right for us.
There's a little fishy.
Oh,
and there goes the fishy.
Oh, there's another one.
These cool patterns, man.
Yeah, you can really see how localized these current variations are.
Quit moving. Okay.
Makes you wonder if the fish hang out in some sort of, I don't know, vortex or something, like an eddy like they do in a stream.
Yeah.
Yeah, very cool.
Maybe that's where they go to get the massage or something, the eddies.
It'd be interesting to put down a time-lapse drop camera that just takes an image like once a day to see how these how and if these sand ripples change over time.
Yeah.
So we have drop cameras on board?
Yeah, we do. Is it a shrimp? Yeah.
So back row, kind of the same drill that we've been going for the past few days, but we're around 1485 meters depth. So we'll expect to be off bottom about 2 hours before our recovery time, so we'll be off bottom at 2. Sound good to you guys?
Yeah. Yeah.
Sounds great.
Great.
And as far as navigation, we can just kind of just keep heading in the same direction, essentially.
Sure thing. We know how much you like the flat and sandy, right, Kevin?
Can't get enough.
So many rocks.
The, yep, that's the thing we talked about earlier on a different, is that what it is?
Oh,
I know what you're talking about.
Phytophore?
Yeah, xenophytophore.
Yeah, that's a little xenophytophore. Yeah, if you zoom in on any of these currents, you see little mounds basically all over the place.
Xenophytophore. I'm glad you knew what I was talking about, mostly because you could see it, but I'm going.
I didn't want to be like, that's a shrimp.
It's like that sediment thing.
You know, sand.
Sometimes the words just don't come, you know?
Yep.
Yeah, the number of times this xenophytophore gets stuck on the tip of my tongue.
Every trivia night ever.
I know, right?
I know, I was just thinking about that the other day.
And that time we all played Pictionary, oof, I was so embarrassed.
Next, xenophytophore random fact. One of the things that's a defining characteristic for them is they excrete barium sulfite crystals.
Wow.
No one really knows why.
Wow.
Barium sulfite crystals?
Yes. These little metal crystals.
Oh, metal crystals out of their bodies.
Mm-hmm.
Come from the sand?
I'm not sure exactly where they come from, but they're known to just like, yeah, excrete these little like barium sulfite crystals.
Super specific, because barium's not even common. It's actually quite rare in this environment.
That's strange. So it's like just like an export for them?
I think so. Although I am not an expert in the biology of xenophyophores. That's actually just a fact I learned about them recently.
Oh, yeah.
Same here.
I was learning about the paleodictyon, which no one knows if it's actually a xenophyophore, but they don't have the barium sulfite crystals, so that was one of the things that makes it a question mark.
Is it a fossil?
It is a fossil, but one that they have found the same textured pattern in the seafloor around hydrothermal vents in the Atlantic, so it seems to still be extant, but it's known all the way back to the Ediacaran.
Wow.
So where's barium on the periodic table?
I think it's an alkali earth metal. It's going to be on the left side.
Okay.
Let me double check.
Group 2.
Do you like any of these rocks?
Yeah, alkaline earth metals. So it's similar to strontium and calcium. So it's a 2+ cation, and no thank you on the rocks. So it can substitute for calcium in carbonate, so that's where it's getting it from, but it's still not very abundant. Has a much larger ionic radii, so it doesn't really like to get into carbonate that well, but it can.
Dead sponge?
Looks like we're getting too much Norbit data. Just got a warning.
I think that's another little black coral.
I'm having a hard time making my eyes see what's happening in front of us. Is that like a big pile of rocks, or is it just spreading out into the distance? I mean, is there elevation there?
No, it seems like there's a little elevation. It's hard to tell.
Looking in the still cam, it looks like there's a little elevation. It's like a little hummock.
Okay.
It's a little anemone on the bottom.
Anemone zoom. Coming wide.
Just kidding. I think so.
Yeah, Kevin, the reason I asked that question about the...
Oh, there was just a tinaphore in the Atlanticam. It was beautiful.
The reason I asked that question about the periodic table is because tunicates, weirdly, are an animal that actually like bioaccumulates vanadium.
Really?
Yeah, so they have a weird, well, I guess it's sort of an exterior covering called a tunic. It's made largely of cellulose, which is really weird for an animal, and which is the same stuff that you find in plants. Gives plants their flexibility, and it is like really pretty highly concentrated, and we think it's essentially like an anti-fouling like paint. It's a toxic heavy metal
Yeah.
that they put into their bodies to keep other stuff from growing on them, at least in shallow water.
Yeah, that's really cool.
That's clever.
Very tall. I'll take a zoom.
Nice big single stalk bamboo there. Looks like it's had some predation.
You can clearly see the nodes on this one.
Probably by a sea star?
Possibly.
I feel like sea stars are usually more thorough.
Yeah. Something took the tissue off right in the middle there.
Are there any fish that eat sea stars?
Any fish that, I have not seen or heard of fish going after sea stars. They're not particularly edible. There are crustaceans that will absolutely eat sea stars.
Yeah.
Mantis shrimp, Dungeness crab.
What's that? Sponge?
Yep, that's a sponge.
It's not a polyapogon, is it?
I do not believe so.
I'll take a zoom. I lied. I don't want zoom. Take it back. Take it back. Take it back.
Make it stop.
Is that one of the sacocalix?
Okay.
Yeah, that may be a sacocalix that just has a really short stalk. It could be also an unstalked euplectellid.
Chris, what's your current speed at right now?
0.2, or no, 0.3.
Oh, you're on. Very nice.
Because past Meredith apparently hates future Meredith, once we get out over this flat sandy bit with the sea pens, can we fire a Niskin?
Yeah.
Process some sandy background.
I would say that present Meredith just believes in future Meredith.
Well, past
Believe
Meredith
in the
and
science.
4 a.m. Meredith will have a conversation
This
about how long they want to be filtering water.
This isn't a problem for your grad students?
No.
Really?
I'm the one who'll be filtering the water today.
Oh, well, today, sure.
Do we have a zoom on that? What is that on the right? Oh no, that's just a dusty rock.
Oh, Roger.
We're getting a little desperate here, aren't we here?
Okay, I will rack back.
Roger. Bender is off.
Raj.
Power is on. Waiting on comms. We're going for 5 and 6, correct?
Correct.
Yep.
Awesome. Still waiting on comms. Got comms.
Roger.
All right.
I'm just going to put it in autos, I think, yeah?
Sure.
Okay.
You might need to cycle the depth because sometimes that one will slam you into either the ground or not.
Okay.
Fun features.
See what happens. I am full racked back.
Roger.
Green or white?
Roger. Go ahead and push on in a wee bit there, please. That's good. You look a little down. There you go.
Trigger.
Right.
Thank you.
Yeah.
Come wide.
Was that 5?
Yep.
Yes.
Sweet, thanks. And that was sample 162.
162.
Okay. Let's get Good job, girl. I didn't do anything. You just were like, buttons, buttons. Manip, Niskin, getting going.
Just doing what you do.
You good, girl. You're good. You usually do this and the buttons. You get to them fast. I swear it's the touch screen.
Yeah. That's infinitely faster.
Yeah.
Are we going to want any other samples before I put away the manip? Anytime soon?
I'm good.
Great. Sounds good.
Yeah.
And Kevin doesn't want to be in this sandy spot, so.
Yeah, yeah. Nothing we need from you.
Roger.
I just want to see Chronocops and maybe some octopus.
Yeah.
Come down a little bit.
If it's gotta be sandy, we might as well find some cool
whale fall.
Yeah.
Yeah.
Here's a message in the chat. Is the CAD model of Hercules available to the public?
No.
Have a zoom on that guy.
I'm trying. I had to think for a second if it's in any open source
I don't think
repositories that we have relative for any of the software stuff, but no, I don't think it is available.
Can you make it available?
There is instructions to construct a Lego Hercules.
Ooh.
No.
What is going on there?
Looks like a shrimp.
I think it's a shrimp.
Oh,
I
it's
think
a
so
shrimp.
too.
Shrimp
It's just that
behind the seapen.
Oh, okay. I thought it was on the sheet then. Never mind.
I think they're hovering together there as buddies.
Yeah. I smell a Pixar movie.
Somehow I just don't think that the seapen is going to be quite as charismatic a story.
No.
Video
The
zoom.
best Pixar movies are when it's a single frame, never moving.
Shrimp, maybe.
That's a cool looking shrimp there.
That's a weird one, yeah.
Okay, come on.
Something broke, Chanel.
She's picturing that shrimp speaking in Finding Nemo voices.
We're only halfway through the cruise. I mean, you need to pull it together.
That's coming from us.
Yeah.
Climbing it like a tree.
Yeah, I thought it was.
And that was what broke Chanel.
We all have our limits.
Oh, yeah. Hey, Jess.
Yep.
If I turn to port,
Sure.
do you want to get a zoom on where the thing is on the thing?
Sure.
Yeah. Get some good visual.
Looks pretty secure where it is.
Okay.
I'm going to go ahead and push on a wee bit there on Atalanta, please.
I'm going to drive forward a little bit.
Roger.
In the still cam view, it looks like Hercules is running around with some kind of Zelda sword.
Yeah. It's ready to defend itself.
Go ahead. You have any more zoom there?
Well, and ready to embed it in a rock. So some true king shrimp can pull it out someday.
King shrimp.
I guess that's a pretty difficult thing to see, yeah?
Yeah. I think it's, yeah, hard to see. That's a cool shot.
Oh, okay.
Follow you around.
Is that a bamboo coal?
Yep, another little single stalk bamboo.
Gotta come partial
Seem to
wide
be
there.
scattered
come
here
on,
and there.
Made myself dizzy.
Is that ever a problem when doing things like removing wraps?
Yeah, it's like sometimes we'll tell science to like close their eyes. Well, I'm closing my eyes. Or like when we do the car wash, that's a little bit nauseating.
What is?
The car washes.
Oh, Reg.
Like if we accidentally dump down in the mud and then we have to like pick up and do some like wagging of the vehicle body to get this mud off, that can be a little bit stomach jostling.
The always inevitably sticky mud from the bottom. It's always stickier than it looks.
Yeah. It's like all of a sudden your ballast is way off. Those are buttons. My auto chocolate's not working. No, I lost a button. Okay, Kylie, get serious.
Good luck.
why start now, right?
Yeah, I mean, there's still 2 weeks left.
We're only an hour into our watch, guys.
Oh.
Got a whole blue water left.
I know, but nothing funny ever happens on blue water. There's no opportunity for auto chocolate jokes then.
Another fish on the left there.
Yeah.